microtest, if that's all, technology industrial finance. Welcome.
Thank you, Rico. Hello, my dear new friends and old friends. Thank you for coming. Today, I will walk you through a complete scientific journey from a timeless Chinese philosophy of life to a practical solution for substantial improving nitrogen use efficiency. This journey is called the GiP Framework, and it originates from China. Let me start with a fundamental question. What is life? Embracing the timeless Chinese concept of Tianren Heyi, harmony between heaven and humanity. Life and its environment, at least from a Chinese perspective, are believed as one inseparable entity. In this context, life is a continuous process of energy and materials exchange, mediated by ions and molecules. The concept of unity of heaven and humanity is indeed a timeless wisdom. Yet, to grasp its relevance in our ever-changing world, especially nowadays, I have dared to add a new variable into this equation, which is time. Then, life can be expressed as a function of both environment and time, a dynamic interpretation of that ancient enduring truth. Then, for a crop, it becomes crop is a function of soil, water, climate, etc., and time. And of course, for this event, soil can be defined as the following. So this is not a metaphor. This is a scientific philosophy, and it is the root of everything we are going to discuss today. If this philosophy is correct, then it leads to an inevitable conclusion. Traditional methods, like measuring nitrogen content in dried leaves or gene expression in disrupted samples, giving us only static snapshots. They miss the dynamic movie of life. So to truly understand how a root absorbs nitrogen, we need to measure the fluxes. I'm so glad today we came across a few excellent works, like Dr. Di from the Chinese Academy reviews that. So the real-time influx and efflux of ammonium, nitrate, proton, potassium, calcium, oxygen, hydrogen peroxide, so on and so forth, right at the root-soil interface. So the question becomes, do we have a tool to do that? The answer is yes. And the tool is AMT, which I have been working for more than 20 years. So AMT stands for non-invasive microtest technology. It derived from vibrating probe technique pioneered by Dr. Jeffy. AMT has become a reliable commercialized system with a publication using it every 2 days. It uses selective microelectrodes to measure ionic molecular concentration gradients near a living sample, then calculate the flux. With AMT, we can finally see the dynamic conversation between a root and its soil, or vice versa. But a tool like AMT is not enough. We need a framework to organize the data into something comparable, shareable, and predictive. That framework is called ammonics, short for ion molecule omics. It is a new paradigm. Ammonics extends the central dogma of biology. DNA makes RNA, RNA makes a protein, and so on and so forth, right? That used to be. But now, protein, together with the environment, produce the ammon, which is the dynamic flux pattern. And the ammon, in turn, produce the final phenotype. In simple words, ammonics turns scattered empty measurements into a structured language. It records not just what the ion molecule moved, but where, when, how fast, directions, and what environment, and in which genotype. This is in vivo functional omics. To make omics truly global, we launched the Global Ammon Project, GiP. GiP is an international alliance, of course, initiated by the Zhongguancun, that's a high-tech park in Beijing, where my company is located. Zhongguancun AMT Industrial Alliance and the AMT International Alliance located in Massachusetts. Our chief scientist is Agaptian Tianren, a legendary plant scientist. And she's also one of my former graduate advisor committee member. GiP's mission is to build a distributed network of research stations that all follow the same standards, along with a shared and AI-ready database. Next, let me show you, so far in the past 20 years, what GiP compatible data already reveals. There are three clear lines of evidence. First, genotype differences. Different rice or corn varieties show distinct ammonium and nitrate flux patterns. That means AMT can directly screen for nitrogen-efficient varieties of course, not only plants, but also soils. Second, environmental response. Fluxes change within minutes when pH or salinity changes. This proves that nitrogen uptake is not a constant. It's not a fixed characteristic. It is a dynamic adaptation. Third, ion coupling. This paper reveals ion coupling. There are other publications you can look up on the website. Also shows ion molecular coupling, molecular coupling, and so on and so forth. So ammonium uptake is tightly, in this paper, linked with proton efflux. Nitrate uptake is linked with potassium and calcium fluxes. So nitrogen, folks, never works alone. So when you put these three lines together, using ammonics as the organizing framework, they converge into a network model. And that model becomes the basis for prediction. Now we come to the application. Smart fertilization means feeding the crop or the grass for the peaches, for the soccer peaches, right? Exactly when and how much it asks, not on fixed schedule. But how do ammonics and the GiP enable this? Fundamentally, ammonics connects molecular regulation to phenotype. We then use AMT to sense real-time root demand. Ammonics and AI transform that data into a dynamic model, which in turn drives variable rate time-specific fertilizer application. This is not science fiction. It is the logical consequence of our founding philosophy. Life is a function of both environment and time. Let me summarize the entire GiP value chain. Life is environment philosophy, MT technology, one paradigm, ammonics, which extends the central dogma, one global project, GiP, an open international alliance, and one solution, smart fertilization, that can raise nitrogen use efficiency theoretically from the current global average of 37% to over 70%. So we would like to thank the Chinese Ministry of Science and Technology and the National Nature Science and Foundation of China, of course. Most of all, we are deeply indebted to our MT collaborators and users worldwide. We also would like to thank Dr. Shi. He has a family emergency, but I'd like to thank the organizing committee and the World Congress of Soil Science, giving us this opportunity to share our journey from the philosophy to the cell function and eventually to the field application. So now it's open with some time schedule like you see here. So please join us. Join the GiP, and thank you all for your attention. And I'd be happy to take some questions. Thank you.
Thank you very much.